Hello, everybody. Uh, Welcome to our ICM virtual seminar in computer and computational science. My name is Marek Michalewicz. I'm the director of Interdisciplinary Center for Mathematical and Computational Modeling, University of Warsaw. We have run a conference entitled Supercomputing Frontiers Europe for the last 6 years. The last edition was in March this year, already during the lockdown time. And so we had to very quickly switch the mode of our conference from personal attendance to virtual. And since we already had a captive audience, and sadly we established a very interesting network of interested people, students, academics. So we decided to continue, change the format, and have regular seminars under this title. So let me first acknowledge some of our partners. Our honorary patron is National Information Processing Institute. We have several media partners, Datanami, ITWiz, HPC Wire, Sztuczna Inteligencja, Enterprise AI, and Computer World. And I have a second slide. Just for those of you who are here for the first time, if you're interested in any topics that were presented in the previous 10 lectures, you are welcome to browse to our website, and all the lectures are recorded, so you can listen to them at your leisure at the time that suits you best. And I really encourage you to check the titles. They cover very wide ground of computer science, computational science, and all of them are really exciting. The last one was given by Stephen Wolfram, the famous creator of Mathematica and Wolfram language and various other things. But every person on this list is distinguished in one way or another, his own domain of expertise. And we already had Professor Michael Bussmann, who is a working colleague of our today's speaker. And with that, maybe I will introduce our today's topic and the speaker. I think on the next slide, please. So we have a great pleasure of welcoming Professor Ivo Sbalzarini from Technische Universität Dresden and several other institutes, including Max Planck Institute of Molecular and Cell Biology and Genetics in Dresden. And let me read here a few sentences. Ivo Sbalzarini is also director of the TUD department in the Center of Systems Biology in Dresden, chair of scientific computing for systems biology on the Faculty of Computer Science at Technical University Dresden. He's permanent senior research group leader with the Max Planck Institute of Molecular Cell Biology and Genetics. He graduated in mechanical engineering from ETH Zurich with specialization in control theory and computational fluid dynamics. He completed his doctorate in computer science at ETH, again, winning several awards for his PhD, Willy Studer Award for further studies, Chofras Award from Weizmann Institute of Science. And at his doctorate work, he pioneered the close collaboration between biology and computer science. After staying at Stanford University in 2000 and NASA Ames in 2002 and Caltech in 2005, Ivo was named Assistant Professor for Computational Science in the Department of Computer Science at ETH Zurich in 2006. In 2012, Ivo and his group moved to Dresden, where he became one of the founding members of the new Max Planck Center for Systems Biology and the Technical University Dresden, chair of scientific computing for systems biology. He also serves as a research avenue leader in the DFG, Cluster of Excellence, Physics of Life, dean of the International Max Planck Graduate School in Cell, Developmental, and Systems Biology, and vice dean of the Faculty of Computer Science at Technical University Dresden. And Today's topic is computational development biology. I can assure you I've had a great pleasure of listening to two talks previously of Ivo, and each time it was such a enlightening and pleasure to learn from Ivo. So I would like you all to have this experience today at our lecture. Ivo, the screen is yours. Thank you.
Thank you very much, Marek, for this generous introduction and for having me on the seminar today. Can I be heard all right and seen? And can you see the slides? Okay. If there's anything going wrong that we lose sound or you cannot see the slides or anything, please just speak up, because I cannot see the chat while I'm sharing the slides. So just feel free to interrupt me if there's any problem. And otherwise, I suggest we collect the questions in the end when I can see the chat and can read what everybody is writing there. So Marek has given a very long introduction, which was very generous. I don't need to say much more, but I want to start delving right into the biological problem that we are studying at the Center for Systems Biology and actually in the Max Planck Institute altogether. We want to understand how cells self-organize into functioning tissues. It is, if you think about it, a bit of a miracle that tens of thousands or millions of cells come together and form a functioning tissue, that they form the right structures, the right shapes, of an organ or a complete organism. And we study this problem by looking at the early embryogenesis of model organisms such as the fruit fly, which is a very popular model organism in molecular and cell biology. What you see on the slide here on top, I hope you can see my pointer, I'm using a big black pointer here, is a microscopy picture from the lab of Lars Hufnagel at EMBL in Heidelberg, where several gene transcripts have been labeled fluorescently. So what you see here is that some cells in the embryo have the blue gene on, some cells express the orange gene, some cells express a green gene. And I'm not going to go into any detail what these genes are or what their chemical names are, because that's not important. What is important is that to take pictures like this, you need to kill the embryo and fix the sample. If you do not fix the embryo, then we have microscopes nowadays that produce videos like the one you see here on the bottom. I hope you can see the video moving, of the three-dimensional development of the tissue, of the embryo, the fly embryo in this case, in space and time. And you can see, you know, here, for example, the neck forming. This part is going to be the head. You can see the mouth forming here. This is going to be the mouth of the fly. This part back here is going to be the main body. It will segment. Eventually, out of the segments, the legs will grow and so on. If we start the movie again from the beginning, you will see that initially, the cells that express the blue gene fold inward, and the green cells push around the envelope of the embryo until the two orange bands have met. And once the two orange bands meet, the folding stops. And so one can ask questions like, how does a blue cell know which way is in and which way is out? The embryo roughly has the shape of an ellipsoid, so the curvature is convex. So if it is simply a phenomenon of buckling, then it would always buckle outward. Why does it buckle inward? How does a cell in the orange band know when it has met a cell from the other orange band and not a neighbor from the cell band? And how do the green cells know which way to walk, such that they push the embryo close and walk around the back and not the wrong way around? So these are simple questions, but the answers to them are not available. We don't understand it. We don't understand how cells do this. What we do know is that cells execute programs. Every cell can be considered as a little chemical computer that executes genetic programs, gene expression programs, that takes decisions, executes also signaling networks, or things like endosomal sorting, which is depicted here in this cartoon, the process by which cells take up molecules from the environment and then sort these molecules. And some molecules go to the nucleus, some molecules are recycled back to the cell membrane, some molecules are degraded. How does the cell know what to do with which molecule? How does this chemical computer operate? And how do millions of these chemical computers communicate with each other by chemical signals, such as morphogens or hormones, but also mechanical signals? Cells can both exert and sense mechanical stresses, and this has been shown to be very important for determining their behavior, or direct cell-cell contacts, which they can change also by moving around. The video you see here on the bottom, by the way, is from the lab of Elliot Meyerowitz at Caltech, which shows how cells in a plant should communicate with one another in order to decide which cells are in the center and are going to grow the flower. So in the language of computer science, what we have in a developing tissue is a massively parallel stochastic chemical computer with a long-range communication network with time-varying topology. And we're talking about a computer here that has millions or even billions of processing units, each of these processing units containing hundreds of billions of molecules or proteins that can store information. So we are talking about the computer system that is orders of magnitude more complex and more powerful than even the fastest available HPC machines on the planet. Yet we want to use the HPC machines that we have to bring some light, to shed some light onto how this biological computer works, what algorithms cells execute when they take decisions, for example, about moving or about dividing. And in order to do that, we follow the classical method of science, which is a combination of observing the system and modeling the system, and then showing in simulations that the models are sufficient to explain the phenomenon. So the first step is observation, where we are really fortunate to have now advanced optics to a point where we can image entire embryos in 3 dimensions plus time at subcellular resolution in movies like the ones you have seen before. These movies are extremely rich sources of information because we can see what every cell is doing at any point in time, and we can see it in the context of all the other cells. The problem is that looking at these movies itself is technically challenging because we require visualization that runs at almost 2 gigapixels per second for real-time rendering of these data. What you see here, by the way, is a fish embryo, another model organism that is frequently used, and it is visualized in a virtual reality cave with projected walls and projected floor. And you can see here the blood vessels of the fish, how the blood vessels develop when the vasculature grows, and the blood cells, the red blood cells, as bright dots that are going to move through these blood vessels. And you can really follow, you can go inside, walk inside the embryo and follow these motions. The microscopes produce images at a rate of a few gigabytes per second, depending on how many cameras are on the microscope. Our microscopes typically produce about 5 gigabytes per second. And we want to stream this data also into a HPC system where it is analyzed in real time. And by analyzed, I typically mean segmented. So what we would want to do here, for example, in the fish, is that the network of the blood vessels is segmented out of these 3D images, so we get a geometric model of the network. We know what the tube lengths are, the diameters, and so on. And we can then use this geometric model to run simulations, for example, of blood hydrodynamics to predict how the blood would move in this network under a certain hypothesis. We can then, of course, calibrate the simulation such that the speed of motion that we see in the images, how fast the blood cells move through the network, and the speed, the velocity field predicted from the simulation are matching. And then we can use the simulation to measure also other things like pressure, for example, or shear stresses on the wall of the blood vessels, which are not directly observable in the image. In order for this to work, we need the simulations to run fast, and this typically means less than 1 second of wall clock time for a simulation time step. So we get, we can overlay the results and we can achieve interactivity. For all of these 3 arrows, we are developing algorithms and implementing them in open source software. For real-time visualization, we have a software called Scenery, which I will talk more about later. We have a software for real-time computer vision, real-time image segmentation, image denoising, and so on. And we have OpenFPM, which I'm mainly going to talk about today as a platform for highly scalable numerical simulations. Together, we can then combine these technologies into a classical reverse engineering loop, where we start from a real-time observation of, again, a fly embryo on this slide. We have a computer simulation of what, in this case, the velocity field of the cell movement would look like under a certain physical hypothesis. And we can use AR and VR technology to superimpose the simulation results with the image data and basically check whether they explain the biology or not. We still cannot automate the comparison because we don't have a good metric for biological correctness. Even 2 genetically identical embryos will behave slightly differently. And of course, a crude physical model will never be exactly like the living system. But what we want is that the simulation captures the biological essence of the process, so that the timing of the developmental events is correct, that dorsal closure has the right directionality, and so on. And this is still best done by looking at it and have a human say, this fits or this does not fit. And this binary information about a model being acceptable or a model not being acceptable can be fed into a machine learning algorithm, which solves the problem of design centering. It does not solve the problem of optimization like typical machine learning methods do. But this design centering algorithm will then tune the parameters of the model such that the scientist says it fits, and it has the highest possible probability of still fitting for the next embryo, which has not yet been imaged. And another thing we can, of course, do is we can directly feed the image data into a machine learning algorithm and ask the question, what would be an appropriate partial differential equation to describe the dynamics that we've seen. Now, all of this, of course, requires high-performance computing. We are talking, as I said before, about several gigabytes of data per second. We're talking about computer simulations with millions or billions of unknowns. And we are basing the entire workflow on one software framework, which is the OpenFPM library. But before I go into details about OpenFPM, which is what I thought was most interesting for the audience today, I want to give you one example of a biological problem that we are working on together with Elisabeth Knust, who is a developmental biologist here at the Max Planck Institute. What you see in the video here on top is a process called dorsal closure. This happens right after what you've seen before. Before you have seen how the tissue folds inwards. And after the tissue has folded inward, the embryo closes the hole, in a sense, and the cells inside delaminate to form the interior organs, whereas the cells outside will go on to form the external organs. And this is a very important process during development. If this process goes wrong, then the embryo cannot differentiate between internal and external tissues and is eventually going to die or have severe birth defects. One such mutation that causes such a defect is in a protein called crumbs. And if you mutate the amino acid at position 10 in the crumbs protein, then instead of getting this beautiful dorsal closure that we see here on the top, you get this more or less chaotic, uncoordinated motion of the cells, and the back of the embryo is not closing. Now, one question that one should immediately ask is, how can this single point mutation on the molecular level have such a drastic effect on the macroscopic phenotype of the tissue? What is the physics? What is the connection? What's happening here? How does, this one amino acid that is in this protein lead to something that has an entirely different behavior? And so this is where biophysics comes in, and where you ask the question, okay, what is the material property of the tissue? What is an appropriate mechanical description of the tissue? What is an appropriate hydrodynamic description of the flows that we are seeing here? And how is all of this regulated and controlled by the molecular signaling and by the molecular pathways where this protein is involved? And one of the most successful mechanical theories for biological materials has been the theory of active polar gels. Active polar gels were proposed by Jacques Pro in Paris, Frank Jülicher here at the Max Planck Institute for Physics in Dresden, Jean-François Joanny in Paris, and also Carsten Kruse in Geneva. And it is a mechanical theory of living materials. Now, the main difference between dead materials and living materials is that living materials can move and deform by themselves. Okay, they can consume a chemical fuel, food, and by consuming this chemical fuel, they can start to move and deform, change their shape, they can grow, they can shrink, and they do this without, they do this actively, intrinsically, and not simply in response to an external force poking them. And so how this happens in cells and in tissues is that there is a network of polymers, filaments, such as actin filaments or microtubules or laminin filaments. There is many different molecules that can form such filament networks in living cells. And on this filament network, you have motor proteins, like myosin motors, for example, for actin, or kinesin and dynein motors for microtubules. And these motor proteins, they consume a chemical fuel, ATP, and they walk on the filaments. And so they hydrolyze this chemical fuel and they convert the energy into a mechanical stress. They can hold onto two filaments and they can slide them against each other. And so there is a set of partial differential equations that is known as the theory of active polar gels, which describes exactly the behavior and the properties of these materials. It's called active because they move and deform by themselves. They have mechanical activity. It's called polar because the materials are anisotropic. They contain filaments that in each position of space give a polarity to the material. And it's a gel, which is something between a solid and a liquid. It's a viscoplastic fluid that behaves elastic on short time scales and fluid on long time scales. And mainly in this theory, you have 5 fields. You have a polarity field, which tells you at each point in space what the orientation of the filaments at that point is. You have a density field, which tells you at each point what the density of the filaments is. You have a chemical potential field, which tells you at each point in space at what rate chemical energy is converted into mechanical energy. You have a velocity field that tells you at each point in space the direction and speed of the movement of the material. And you have a pressure field that tells you how pressure is, hydrostatic pressure is distributed in the material. And of course, you can ask the question, if I know the polarity, density, and chemical potential field of a tissue from an image, because in the image I can segment these filaments, I can see how dense they are, what the orientation is. Can I solve these PDEs to predict, for example, the velocity? To predict, with this arrangement of filaments and this distribution of motors, how is the material going to deform into the future? What shape is it going to attain? And it turns out that numerically solving these PDEs is exceedingly hard. These PDEs are highly nonlinear. They contain fourth derivatives. They are fourth or sixth order nonlinear, coupled PDEs. And many standard numerical methods failed. And I think we understand now why they failed, and we were able to produce a method that is able to simulate these PDEs or to solve these PDEs numerically based on particle methods. And I will give you an idea of what particle methods are and why they work in just a minute from now. But applying this simulation or this theory now to our problem of dorsal closure, we can take some cells out of this contracting tissue, and we can simulate just one tiny piece of the cortex of one of these cells. And we can say inside of the cell we have cytoplasm, cell plasm, which we model as a highly viscous but passive fluid. We have under the surface of the cell a layer of an active fluid, which is consisting of actin polymers and myosin motors that has a polarity field and generates a velocity field. And at the outer edge of the cell, we have the plasma membrane, the cell membrane, which we again model as a passive fluid. Between these fluids, there is no hard walls, but these fluids swim on top of each other, in a sense, like oil swims on water, and there is friction between these different layers of fluids. So if we take this system and we solve the equations of active polar gels in the light blue area, and we solve the classical hydrodynamic equations for passive fluids in the dark blue and in the red area, we can, for example, compute the Lyapunov exponent of the flow. The Lyapunov exponent tells us whether the flow is stable, meaning if I perturb it a little bit, will it go back to what it was before, or will it explode and start doing something completely chaotic upon a small perturbation? So negative Lyapunov exponents mean the system is stable. So if I apply a small perturbation, it will go back to normal. And positive Lyapunov exponents mean the system is unstable and it will become chaotic. What we found, if in the simulation we increase the activity of the motors, there is a region where nothing moves. The material is static. And once you overcome this, then you go into a region where you start to see flows. Nice laminar flows that are closing the gap, like you have seen in the video. And this is similar to stick friction, but now it's in an active material, but you need to have at least a certain level of activity, a certain availability of fuel to make the material move at all. If you increase the activity even more, then you start to see traveling waves and vortices, which are classical hallmarks of borderline stability. And if you increase even more, there is a nonlinear phase transition into chaos. This nonlinear transition cannot be found by linear stability analysis, but we found it in the numerical simulations. This is some visualizations. This is what the laminar flow looks like. The tissue is just, the cells are all just moving in one direction very nicely, like we see in the healthy embryo. This is what happens if you increase a bit the activity, and this is what happens if you increase even more. So now, of course, the hypothesis is that what we saw in this Crumbs mutant was this type of chaos, and this type of chaos, by the way, is called active turbulence. It's different from inertial turbulence. It has a different scaling law, and it has different hydrodynamics altogether, but it is chaotic and aperiodic, and it does have a power law spectrum, but with a different exponent. So physicists call this active turbulence. And what we can now hypothesize is that when we mutate this Crumbs protein, we are disrupting a regulatory pathway that keeps the activity of the motors in the cells in the healthy area. And when we dysregulate the activity basically shoots up and the tissue goes into active turbulence. So these are some sort of phase transitions that we can find in numerical simulations that are impossible to detect experimentally, because you never know what you're looking at, and that also are impossible to detect using analytical work. And we do these simulations, as I already mentioned, using particle methods, and this is why these methods work. Particle methods are numerical methods to solve PDEs, where the solution is discretized on particles. Particles are points in space that carry the solution value. And in our case of active polar gel, the particle also carries the polarity. And then particles have two properties in our simulation. The particles move with the flow. We say they are Lagrangian discretization points. So if the flow deforms the material, the particles of the numerical discretization, they move with the flow. This removes the nonlinearity of the advection operator from the equations. And the particles also have a polarity vector that, if it moves with the flow, changes, of course, its direction, but it does not change its length. And this turned out to be the main problem with previous numerical methods. The polarity vector is a unit vector in the theory. And if you make a small numerical mistake and it deviates a little bit from a unit vector, then this mistake will get amplified to the power of 4 into the velocity equation, and the velocity will become wrong, which again feeds back into the polarity equation. So you really cannot make any even small mistake on the magnitude of the polarity. And doing it with particle methods that are Lagrangian, of course, satisfies that exactly. But if you try to do finite elements or finite differences, it will fail. We still need a mesh from time to time, because the particles could move to locations where they're not needed, or we could have areas where the particles vanish. And so from time to time, we interpolate the particles back to a regular mesh and restart from there. This is a step called remeshing, and this basically makes sure the simulation remains well resolved and well behaved. In addition, we can use the mesh to do things like Fourier transforms to solve Poisson equations, which is more efficiently done on the mesh than on particles. So taken together, the particle methods for fluid dynamics, not only active gels, but also classical Navier-Stokes equations, particles can do two things. They can move with the flow, and they can interact with each other, for example, to do diffusion. We solved the hydrodynamics on the mesh, and for this we have moment-conserving particle mesh interpolations that conserve the mass and the impulse of the system exactly. But particle methods turn out to be more general than just fluid dynamics. We can actually see them as a unifying computational frameworks. Particles are just points that have a position in some space and properties that they carry. The space can be physical space, but the space can also be phase space, or the space can be parameter space of a machine learning algorithm. And if I take this more general view of particle methods, then I can see that they can be used to simulate models of all 4 types. They can be used to simulate deterministic continuous models, which are typically formulated as partial differential equations. In the way I described before, particles are discretization points that discretize these continuous fields. They can also be used, obviously, to simulate discrete deterministic models, such as molecular dynamic simulations, where the particles can directly represent discrete entities, such as atoms, or in coarse-grained molecular dynamics, maybe whole molecules, that interact with each other according to deterministic force fields. Particle methods can also be used to solve stochastic differential equations, which are continuous stochastic models, in much the same way as you use them for PDEs, but now the interactions between particles involve randomness. And of course, they can also simulate stochastic discrete models. And the famous example for that are agent-based simulations, where particles represent, for example, animals in an ecosystem or cars in road traffic. And they interact with each other according to stochastic rules. So the interactions between particles can be either deterministic or stochastic. So with this, we can do things like this one here, where we simulate a deterministic continuous model that tells us how a biological membrane would basically divide into 4 pieces as driven by a Turing-type reaction diffusion system that goes on its surface. We can simulate models like this, which is a discrete deterministic molecular simulation of how motor proteins diffuse around in the cell and then eventually bind to these polymer filaments and walk along them. We can also use particle methods to do image segmentation. This is an example here where we find these green objects in the image. These green objects are endosomes in cells that process molecular signals. And we segment them. As you can see, we segment them with sub-pixel resolution. The segmentation is represented as a spline, and the control points of this spline are the particles. Or we can use the particle methods for optimization and machine learning. In this case, you see a visualization how a search algorithm finds the global minimum in this very multimodal and rugged cost function. In this case, the particles are simply the samples of the search algorithm, and they interact with each other to find the global minimum. So, viewed as a unifying computational framework, particles can interact with each other in order to either discretize and therefore numerically solve mathematical models. They can do this in arbitrarily complex geometries because we don't need to generate the mesh, so the complexity of the geometry is not the limiting factor. But particles can also directly mimic physical or biological or social interactions between discrete constituents of a system, such as animals or molecules or people, in which case we don't solve a PDE, but we simulate bottom-up some dynamics. We can combine them with meshes. We can combine the particle method with any of the existing mesh-based methods using moment-conserving particle mesh interpolation schemes. And at the end of the day, every particle-based simulation boils down to these two equations. We have a set of particles, P, and for each particle, we need to know how does it move. So what is dx pdt? What is the time derivative of its position? And how does it evolve its property? Omega is just the property of the particle. Can be the charge of an atom, or can be the age of an animal, or can be the pressure of a fluid element. And so in the most general case, both the motion of a certain particle and the evolution of a certain particle depends through some nonlinear function on the positions and properties of all other particles. That's the most general case. That's the classical n-body problem. And so because particle methods boil down to such a nice stereotyped mathematical framework, they are perfect for software engineering. And this is where the OpenFPM library comes in. About, well, I guess almost 20 years ago or 15 years ago, yeah, we started, when I was still a master's student, we started thinking about a software library, a software framework that could be used to implement any type of particle method. And everything you need to do these things down here. So you need time-stepping schemes, you need these moment-conserving interpolations, you need particles to be able to efficiently find their neighbors in high-dimensional spaces, would be pre-packaged in a library, and you could simply use these building blocks to build your particle-based simulation application. This gave rise to the PPM library, the Parallel Particle Mesh library, which was reasonably successful, but started to become old and not using the most modern software architectures anymore. So we decided 6 years ago to replace it with a successor, and that is OpenFPM. OpenFPM was finally published in 2019, or the paper about it was published in 2019. And it is a C++ library that makes heavy use of template metaprogramming and compile time code generation to provide exactly these building blocks that you need for particle simulations, but also mesh-based simulations and any combination of particles and meshes. It also has a numerics module that includes frequently used solvers like Fourier transforms and Poisson solvers and multigrid solvers and so on. Through template metaprogramming, we completely abstract from the hardware, so we can compile OpenFPM for GPUs, but we can also compile it for distributed memory CPU clusters. And on top of this, we have a development environment where people can implement their application clients, for example, for diffusion in complex spaces or graph theory or image segmentation or plasma physics. Or this is a diffusion of a protein in a cellular organelle. The OpenFPM core itself has a layered architecture where just above the hardware we have our own memory allocators. So OpenFPM does not use the memory allocation of the operating system, but it implements its own memory allocators that are faster and more flexible, and you save all the kernel calls. On top of the memory allocators, we have the memory layout model, which basically says whether you store something as a struct of arrays or an array of structs or a linked list or whatever. And using these memory layouts, OpenFPM then defines first single core data structures. And the single core data structures we have in OpenFPM are these three. We can define multidimensional arrays, we can define vectors, and we can define sparse grids. A multidimensional array of dimension 3, where each element of the array is a structure of an int and then a float vector and then a double array that I want to allocate on CUDA memory with a struct of array layout would look like this. So multidimensional arrays in OpenFPM are called grids. So you would say grid, and everything that defines this grid is passed as a variadic template parameter. So the first template parameter says what is the dimension of the grid. The second template parameter says what is stored on the grid. In this case, this aggregate, which is again a nested variadic template. The third template parameter says what memory layout should be used. In this case, we use memory trait inter, which actually stands for interleaved. So we use an interleaved memory layout, which is a struct of arrays. And the fourth template parameter says where to allocate. In this case, on the GPU, in CUDA memory. Same for vector. We can have a vector that carries a vector of aggregates. In this case, we want to use a linear memory layout, which is an array of structures, and allocate it on the heap memory. And the sparse grid has a dimension. It also stores something, in this case, an aggregate of, again, other data types. It also has a memory layout that can be chosen. It also has a location. And in addition, as a specialty for sparse grids, we can also say how the sparse grid should be indexed. In this case, we use Morton Z ordering for indexing the array, and what sparsity it should have. In this case, it has a chunk sparsity with 8 by 8 blocks. And so we say here grid Z Morton blocks, dimension 2, block size 8. And so this is how you define single core data structures, and the way you access these data structures, the iterators that you can use over them, is independent of what the underlying memory layout or what the underlying target device is. This becomes completely transparent and independent from the parameters. Using these single core data structures, OpenFPM then implements domain decompositions and communication subroutines. Domain decompositions, like always in parallel computing, we decompose the domain into subdomains that are distributed across processors. For example, if particles is a vector that stores the particles, then we can use particles.decompose to decompose it, and particles.redecompose to do runtime dynamic load balancing, where the decomposition is not recomputed from scratch, but we take the old decomposition and adjust it. In this case, we are simulating a flow of water in this tank here. And of course, initially, all the particles are here, but as the water moves over here, the domain decomposition follows it. And we implement communication subroutines so that the different processors can communicate with one another. And really, the only thing that OpenFPM knows is asynchronous non-blocking MPI communication of bit streams. OpenFPM does not care about what this bit stream means. So whether it is a grid or a sparse grid or a vector, all of this gets serialized and packed into a simple bit stream, which is then sent as an asynchronous non-blocking MPI call and unpacked on the other side. So if you want a new data structure, all you have to do is you have to implement a serialization and a deserialization method for this data structure, and all the communication machinery of OpenFPM will work transparently. And using these communication and domain decompositions, OpenFPM then defines distributed data structures, which are, well, fittingly called vector dist for a distributed vector and grid dist for a distributed grid, which have yeah, some sort of domain decomposition that you can choose and can be distributed across multiple GPUs or across multiple CPUs. The iterators you can use to now iterate through the particles, they don't know about this. You will just have an iterator and you don't know on which CPU a certain particle is stored, and you don't need to know. And with this, you can implement very compact and scalable simulations. For example, you can implement a smooth particle hydrodynamics simulation of classical fluid mechanics, a granular flow simulation of these grains, of these spheres here drizzling down a plane. This is a biophysical model of a pattern-forming chemical system in cells, the Gray-Scott system. This is a model of plasma physics here, and this is a model of compressible fluid dynamics. And you see that all of these are just a few hundred lines of C++ code. And they all work on CPUs and GPUs. They scale to distributed memory clusters. They have built-in visualization capabilities and so on in just this few hundred lines of C++ code. There is no loss of performance if you benchmark the high-level data structures. So the distributed data structures, VD is a distributed vector, against just plain arrays in C++. You will find that the memory bandwidth we get is the same. And both of them on this GPU here achieve almost 90% of the theoretical bandwidth of the memory of the VRAM. So there's no loss of performance. And this is because, of course, all this template magic is resolved at compile time, and the compiler probably produces something like this. Probably uglier than this, but something like this. And so to make this even more usable for numerical simulations, we put on top a C++ template expression system to express partial differential equations. And here you see an example. This is a simple advection reaction diffusion equation here, where you have an advection term, you have diffusion terms, and you have reaction terms. And if we want to implement these equations in OpenFPM, we basically define first the differential operators. We define an operator lap, which is of class Laplacian, over the particles, and the particles are a vector dist that is defined up here with a certain convergence order. We define an advection operator also over the same particles. And then we can simply, in the time loop of the simulation, write stuff like that. We can write ux equals ux plus dt times advection of ux with velocity v, blah, blah, blah. So this very much starts to look like the actual PDE, that you can simply type them in these expressions. And again, all of these things are template parameters that get resolved at compile time. You can also implement implicit solvers using this methodology. Here, for example, we want to solve this Poisson equation to solve for the velocity given the pressure. And again, you say what numerical scheme you want to use for the solver. In this case, we use DCPSE scheme. Then you define the right-hand sides of the equation that you want to solve. You impose the right-hand sides on the bulk, and you impose the boundary conditions. In this case, the velocity should be 0 at the boundary. You impose the boundary conditions on the boundary, and then you simply say solver.solve for Vx and Vy. And again, this very nice high-level expression. And this solver, by the way, is then just using PETSI or Trillinos or anything that you want to interface with behind the scenes to actually solve the linear system. Again, this does not lead to performance loss. If we benchmark this against state-of-the-art handwritten parallel codes, such as LUMPS for molecular dynamics, dual SPH for SPH, or the AMRX code for finite differences, you see that the OpenFPM generated code either scales the same, so these are all strong scalings for increasing number of processing cores, or even a bit better, or even a bit much better than the state-of-the-art codes. And the cool thing about this whole template expression system is that in order to run now the same simulation on a GPU, I only need to recompile the code. That's all I need to do. I don't need to write any CUDA by hand or any kernels. I can just recompile the code for the GPU target. And in this case, we run the same, water in a tank SPH simulation now on a GTX 1080. And the speed up we got over the Xeon CPU was about 200-fold. So, but now coming back to active polar gels, we want to use this nice OpenFPM library now to actually simulate these complicated active polar gels. Active polar gels, as I said before, consist of multiple equations. One equation is the equation for the polarity, which tells us how the polarity vector, so the orientation of the filaments in space, how that changes over time. That has some component, of course, where the polarity gets stratified by the flow velocity u, and it has some component where the polarity gets rotated by the vorticity of the flow. And it has this mysterious component here, which is the molecular field, which tells us how the motor proteins are pulling on the filaments. We have a force balance equation, which in this case is simply a Stokes equation, saying that the divergence of the stress tensor minus the gradient of the pressure is 0. We have the incompressibility constraint, which simply says the divergence of the velocity is 0. And we have a stress-strain relationship, or a so-called constitutive relationship for the fluid, which tells us how the velocity depends on the rate of strain tensor. This is a bit more complicated because we have all these liquid crystal terms here and all these active terms of the motor proteins pulling. But it still looks kind of, it's more or less a Navier-Stokes equation with 1 or 2 additional terms. You might say it's not such a big deal. But if we just write this out component-wise, instead of using this, beautifully compact Einstein notation, then just the x component of the stress-strain relation alone in 2D looks like this. This is only the x component of the stress-strain relation in 2D. And you see now here you have all these nonlinearities, px to the power of 3, px squared times px squared. And you have higher order derivatives here. So this is not easy to solve numerically. And then in our template expression system, the C++ code for this looks like so. And you see that it is not much more complicated than the actual equations. So the complexity of the code and the complexity of the equations, they are commensurate. And we can use things like dx here, which is the derivative in x, or dxy, which is the mixed derivative in x and y, and simply write these things. And these are all vector operations. So here, for example, pol x is the x component of the polarity field, but over all particles. So pol x is a whole vector. It's a whole distributed vector that is distributed over multiple CPUs. But I don't see that here. And then I can run such a simulation where I use DCPSE to discretize the differential operators. I do a Lagrangian simulation, meaning the particles move with the flow. And so when they move from one processor to the other, they also get communicated over MPI. And I use a pressure correction term to impose the incompressibility constraint. I start from some initial condition for the polarity field, which is some sines and cosines, not important. You see it visualized down here on the lower right. This is the visualization of the polarity field initially. On the left, you see the visualization of the velocity. And on the top right, you see the visualization of the energy, of the Frank free energy of the system. And you also see superimposed here the domain decomposition. So you see here these little boxes. These are the domain decomposition as it is given to different processors in the cluster. And then this is how time progresses. And the flow spontaneously starts to move. It's an active fluid, okay? It does not need an initial impulse. It is at rest initially, and then it spontaneously starts to move. The energy goes up, and the polarity field rearranges. We get the proper convergence. We use second order DCPSE operator. It actually converges second order, both in the L infinity and in the L2 norm, so the solvers are correct. To make things even more simple to use for computational physicists also that do not know C++ template expression systems, we are developing a Python-like domain-specific language together with Geronimo Castrillon, who is the professor of programming languages and compiler construction at TU Dresden, and this we presented at ICM TOMS 2 years ago. which is a language that is really inspired in Python, but it admits using Unicode, it admits mathematical notations for the equations. So you can use here partial derivatives and nabla and Laplace operators. So you can again insert your partial differential equations in more or less mathematical notation. And in order to tell the compiler what numerical method you want to use, you import the corresponding modules. By saying import Runge-Kutta as RK4, you say that the time integration should be done with Runge-Kutta 4. By saying import Laplace PSE 2D as LAP, you say that the Laplace operator or the operator that you call LAP, whatever you use it then in the end, should use a second order PSE discretization. And then the code to do that will be generated automatically. It will link against OpenFPM and run on your target architecture. And the last thing, of course, we want to do is to visualize this. We do not only want to visualize 3D volumetric images like I have shown you before. We want to visualize also geometric data or simulation results. And we want to visualize these things on screen in virtual reality headsets, so we can see the 3D visualization of the simulation and of the image, maybe even overlaid on top of each other, and in room-sized equipment like virtual reality caves or power walls, where you have multiple graphics cards that drive the different projectors. And for all of this, we have again one software. This is Scenery. And of course, now it gets interesting when we combine Scenery with OpenFPM to do in situ visualization of a running simulation. So while a simulation runs, in this case, a simulation of bile flow in the liver, you see here liver cells and then the network of bile channels and blood vessels. And you can look at the simulation of the visualization of the simulation while it runs. And this is a paper we are going to present actually next month at ISAF workshop in supercomputing 2020 to provide transparent asynchronous CPU, GPU in situ visualization for OpenFPM, where in order to enable it for a simulation, you need to change only 2 lines of code. When you initialize the OpenFPM library, you need to say in situ visualization, so the appropriate code gets generated. And if you have a certain distributed vector that you want to visualize, you simply say vd.visualize. As a template parameter, you say which element of the vector should be visualized. And in parentheses, you can give optional arguments. For example, the color with which it should be visualized, or the type of rendering with which it should be visualized, or whether you want to visualize the magnitude of a vector or a component. And then you see these movies like here. Yeah, this is not the movie, but you see these visualizations like here. Our architecture that combines OpenFPM and Scenery basically runs one visualization process on each compute node. The visualization runs on the GPU, whereas the simulation runs on the CPU. They communicate via shared memory and direct memory access. And the compositing of the images to one complete view is done on the head node of the cluster, which again runs an OpenFPM instance to communicate the MPI messages to the compute nodes, and the Scenery instance to composite the image and encode it as a H.264 video stream. And this video stream then gets sent to a remote client, for example, a web browser, from which you can view the visualization and change the simulation parameters at runtime. This system works and achieves interactive frame rates for very large simulations like 248 cubed on 18 nodes. It was a very large simulation on a reasonably small computer cluster. We still achieve something like 10 frames per second for the visualization. For smaller simulations, we can achieve more than 50 frames per second, which is really a smooth interactive visualization that you can get from this. This gray Scott simulation here, just to give you an idea, to solve for 1 billion unknowns on 240 cores in a strong scaling took about 1 second per time step for the simulation and about 60 milliseconds per time step for the visualization. in our asynchronous implementation. Here we compare with the synchronous implementation like ParaView, just to show that we are faster. And this runs in the CSPT Cave, which is the virtual reality environment here at the Center for Systems Biology, where people can go inside and walk around in developing embryos, look at live imagery from the microscopes that gets also streamed off the microscope in real time or comes from file server, and it can look at visualizations of simulations. The picture here you see are kids. We are using this also a lot for scientific outreach. We have school classes, we had TV shows as guests in our cave that come and experience this. And of course, we had politicians and very famous scientists that also came and immersed themselves into developing embryos and marveled, really marveled at what cells do and how they behave towards each other when they form a tissue. So altogether, this allows us to do research in a lot of cool areas of computer science, from immersive media and AR, VR to high-performance computing to smart devices and control of optical microscopes, and of course, artificial intelligence and machine learning. All our software is open source, and you can find it and download it and contribute to it and join. The software framework we have for image analysis is the Mosaic suite for Fiji and ImageJ. It does motion tracking, image segmentation, interaction analysis, image enhancement, denoising, deconvolution, dehazing, and so on. We have OpenFPM, which I talked in detail today, for hybrid particle mesh simulations. We have the rapid development environment and the domain-specific language on top of OpenFPM, which you can use, but you don't have to. We have Clear Volume, which I did not mention at all, which is a real-time microscope control and data acquisition software. So this is what takes the data from the microscope and streams it into the VR cave. And we have Scenery, which I also talked about today, which is open source virtual and augmented reality framework with user interaction. And all of these great software, of course, and all of these fantastic algorithms were not developed by me, but by people in my group and by people we collaborate with. So I want to end by thanking, of course, all the present members in the group that are contributing on the biology or the computer science or the mathematics of what we do, and also the past members of the group which have laid the foundations for all of this. And of course, we collaborate with many people at the CSPD and worldwide in computer science, in biology, in physics, to make this interdisciplinary fusion. And if you want to know more about our work, if you want to see the PDFs of our publications or get the code, this is our URL, our website, and this is our Twitter handle if you care to receive announcements of talks or visitors or papers that come out from our group. And with this, I would like to thank you very much for being here so numerously today and for your attention.